When the outbreak first stopped the world, one deliberate, deeply curious question began to echo. Did human hands engineer this virus? For months, the official narrative focused strictly on nature. Researchers tracked genetic lineages back to wildlife, suggesting the virus reached humans through a simple, unassisted biological spillover from an animal host. But two separate theories have since complicated that story: the bioweapon hypothesis and the lab leak. In public debates, these ideas are frequently lumped together. In reality, they describe two different events. To a forensic virologist, intentional weaponization and accidental exposure require different sets of biological evidence. Proving one does not automatically prove the other. To find the truth, we have to look past the political headlines, step inside the laboratory, and audit the microscopic evidence directly. Let's start with the bioweapon hypothesis. Building a pathogen from scratch typically requires genome splicing, a method where scientists cut and paste specific genetic sequences to design dangerous new traits. This is a DNA sequencing chromatogram, mapping the base pairs of a virus's genetic code. When humans artificially manipulate this code, it leaves microscopic fingerprints. Forensic virologists scan these readouts for synthetic markers, clean surgical cuts proving intentional tampering. When scientists mapped this virus, those markers were missing. Instead, the sequence looked chaotic and unpolished, mirroring the random, messy signature of natural evolution. Because the virus lacks the structural hallmarks of an engineered weapon, the investigation pivots toward the second, more plausible human origin scenario. The lab leak theory focuses on accidental exposure rather than malice, suggesting a naturally evolving virus escaped a facility where it was being studied. This involves gain-of-function research. Scientists intentionally encourage a virus to develop new traits, like a spike protein attaching more easily to human cells. This work is defensive. By making a virus stronger, researchers can predict pandemics and design vaccines. To conduct these studies, laboratories must cultivate high concentrations of these enhanced pathogens. This image shows blue-tinted viral particles replicating within a host cell. This is the paradox of modern virology. The very act of studying apocalyptic pathogens to prevent a crisis creates the biological material necessary to start one. To understand how that material escapes, we have to look at the physical engineering of biosafety-level containment labs. Researchers working with high-level pathogens operate within a strict, pressurized infrastructure. They wear reinforced suits and remain tethered to an external air hose to ensure no direct contact with the samples. Real-world lab leaks are rarely dramatic. They're usually caused by microscopic, invisible points of failure, a degrading seal, or a hairline tear going completely unnoticed. Over the last few decades, high-security labs around the world have documented dozens of accidental exposures caused by these exact types of human and mechanical errors. You can build the most robust containment facility on the planet, but human error is a variable that cannot be removed. A perfect containment system is a mathematical impossibility. The scientific evidence currently leaves us with a difficult reality. Researchers cannot definitively rule out an accidental laboratory exposure. This lingering ambiguity has changed how we view scientific progress, removing the public's illusion of absolute safety in these facilities. Whether the virus originated in a remote cave or a petri dish is no longer the most urgent question. The reality is that we already have the tools and the geographic infrastructure required to cultivate world-ending pathogens. Until there is a global reckoning on how we oversee this research, the architecture for the next contagion is already built.